Good afternoon. My name is Hsiang-Yun Wu from TU Wien, Austria. Today I will present Metabopolis, scalable network layout for biological pathway diagrams in urban map style. A biological network is any network that applies to biological system. It is also well known as small, meaning that they are highly clustered with shortcuts that connect the clusters. Among biological networks, metabolic networks, which are formed by a set of metabolic pathways, are often studied. However, the size of the network is often large, and readers may get lost easily during the exploration. The image on the right shows a human metabolic network, which is often stored in such a format that describes reactions associated with input chemical component as well as the corresponding product. This map was created by 5 undergraduate biochemists in over 20 months by manually reworking on the layout and fixing the errors based on the information in the latest literature. It is a tedious and time-consuming process. This is because several challenges remain, and thus the problem cannot be easily solved by the machine. For example, preserving mental map of the diagram or customizing the network are not easy. To the best of our knowledge, no good clutter management strategy exists. A readable visual hierarchy is also missing to present low-level and high-level relationship. Initially, we tried to visualize the Recon dataset, while we only see a hairball like this. Since the scale of biological network is similar to road network, we were inspired to introduce urban map metaphor to visualize biological network. For example, like the Chicago map you see here, and the abstraction of an urban map. So now I will quickly show how our system looks like. We consider functional groups as city blocks and chemical components as inhabitants in the city. This clip shows sub-pathways in human metabolism, including 11 categories highlighted in color blocks. The red route here indicates a path for cytoplasmatic oxidation of glucose in cytoplasm in order to obtain ATP. And the blue route here shows how humans transform ammonia to urea in order to eliminate the toxic ammonia in the urea cycle. In this example, the chemical component globally move across multiple categories, from mitochondria to cytoplasma through the transport pathways. Our system also supports interaction that allows users to adjust category. As well as highlighting the chemical component. It has been done through formulating several aesthetic criteria to mathematical equations and solve equations using optimization technique. We use mixed integer programming in our system since it is a well-studied field and can potentially find us an optimal solution. I will not have time to go through all technical detail today, so I would like to give a high-level concept here. First, we use a constraint planar graph as skeleton to arrange the placement of blocks, by assuming those share common chemical components should be placed close to each other. Once we have initial position, we add constraint described previously to our optimization process. This is done by formulating each constraint to a set of linear equations as shown in this slide. Once the blocks are placed, we use tree map to settle our city structure and place subnetwork along the grid for better readability. For each intra-block and inter-block network, we incorporate a network flow algorithm for routing the edges. This is an example of reproducing cake overview pathway. It contains approximately 4,000 nodes and edges. The diagram on the right is computed using Metabopolis. The result is similar to the original cake map, since blocks with warm and cool colors are grouped together as originally designed, you see on the left. The image on the right again show the diagram generated by Metabopolis, which data is from the Recon project. This left figure shows the same data from Recon map. To the best of our knowledge, our diagram is the first automatic approach that still clearly shows the detailed connectivity of chemical components. We therefore successfully reduced the layout time from 20 months to 2 hours. While the current approach has some limitation, it cannot fully achieve the high-quality result as handcrafted by humans, since several pre-knowledge has been collected in designer's map and could not be formulated accurately. This paper presents the very first approach to automatically design biological pathway diagram in urban map style. In the future, we will involve the creation of a visual web repository where biologists can continuously add and create their map together with sharing the map with the rest of the community. So with this slide, I would like to thank you for your attention, and I'm open for questions.